See everyone popping up on the screen. Happy New Year to everyone. I know some of you might have been on our program the other day, but I wasn't facilitating. So this is my first program for 2022 to facilitate. So Happy New Year to all of our PMD Alliance friends. And if you are new to joining a PMD Alliance program, then welcome, welcome. We're so very happy that you're here. We're excited to have this conversation today. And just a bit of housekeeping. My name is Anissa Mitchell with PMD Alliance, and I'm going to be facilitating today with our speaker, Dr. Larson. And before we turn it over to him, I want to just ask you to go ahead and pop open your chat box. Give us a shout out. Let us know where you're joining us from. We are going to be taking your questions from there today. He's going to do a slide presentation, and then we'll go back to those questions at the end. And hopefully we'll be able to get to the majority of them. We will not be able to unmute today. We have a lot of people joining us, but I'm very excited to have everybody here. We certainly want to hear from you. We love to see where everyone's coming from and hear your burning questions that we have. So thank you. I see lots of people from all over New Mexico and Tennessee, Chicago, even Florida. And I am excited to introduce our speaker today, Dr. Paul Larson. He is a professor of neurosurgery at the University of Arizona and chief of neurosurgery at the Southern Arizona VA healthcare system. Now, I'm excited about this topic because he's one of the largest experienced researcher in intracranial delivery of novel therapeutics for neurodegenerative disorders. And we're going to be talking about the biological approaches for surgical treatment of Parkinson's. So I'm very excited about this conversation and very excited to have Dr. Larson with us. So Dr. Larson, thank you so much for joining us. And I am going to turn this over to you because I know you have a very robust conversation to share with us.
Great. Well, thank you guys very much. I am going to share my screen, get some slides going. We love slides. All right, everyone see my screen okay? Kelly's giving me the thumbs up, so I think we're good. All right, great. Well, thanks everyone for joining today. I'm gonna spend the first sort of part of our time talking about biologic approaches for surgery for Parkinson's disease. And then we're going to leave plenty of time at the end for question and answers. So I do now work in Arizona. I just recently left University of California, San Francisco, which is where I worked for many years. I still have a faculty position there, but I don't teach there anymore, don't really operate there anymore. And I do a lot of consulting and research with a lot of companies, and we'll talk about some of these more as we go through the hour. So I give this talk to multiple groups. Some of them aren't as familiar with Parkinson's disease as this group is. So I don't need to explain what Parkinson's disease is to the group this afternoon. This is a very typical patient that has advanced symptoms, a lot of tremor, a lot of slowness, a lot of stiffness. And this is really what we are trying to treat, ultimately what we're trying to cure. And medications early on in the disease can be quite helpful, but patients often do get to a stage in their disease where medications become less and less effective. And we need to find better solutions for patients when they get to this stage of their Parkinson's disease. And two of the major issues that we run into when medications become less effective is what we call motor fluctuations. This means that patients are good right after they take their medications, and we say they're in a good on state, so they can function, their tremor is less, or their stiffness is less, or they can walk better. But then they fluctuate before their next dose of medications. They turn off and their symptoms come back. And then they have to take their medications again, and they're on this roller coaster throughout the day. And we call these patients fluctuators. These are motor fluctuations. The other thing that we can run into is a side effect of medication treatment, and that's something called dyskinesia. So dyskinesias are a side effect of taking the medications such as Sinemet or carbidopa-levodopa for our Parkinson's disease. So up to now, and still currently, the best surgical approach we have to try to treat Parkinson's is this technique called deep brain stimulation. And I suspect that most people on the call are familiar with this. I suspect there are some on the call that have had this operation. But the best way to think about DBS is a pacemaker. It is a pacemaker for the brain. It sends a high frequency electrical stimulation into a particular part of the brain that's involved in movement. And by putting a sort of a regular signal into that circuit in the brain, it can normalize the activity in that circuit, and it can reduce the symptoms of Parkinson's disease. It can reduce tremor, it can reduce stiffness, it can speed up our movements, make our walking better, and it can also help with dyskinesia. So DBS is great. There's hundreds of thousands of people that have now had this, and it's very good at treating symptoms, but it has some drawbacks. It's a symptomatic therapy, which means that it doesn't alter the progression of Parkinson's. It doesn't alter the abnormal brain biology in Parkinson's. And it's what we call a hardware-based treatment. It's a battery pack that goes in the chest. It's wires that go up into the brain. And whenever you're putting hardware into the body that doesn't belong there, you can run into complications. And the components of the system can get infected. They can erode through the skin. They can shift out of position. It's a system that depends on energy. So there's a battery that has to be replaced periodically, or we now have rechargeable batteries, but those have to be recharged by the patient. So there's a bit of a time investment on the part of the patient and their doctors. And it's not an automatic treatment. You know, you have to come in, you have to have the stimulator settings adjusted to try to continue to maximize improvement in symptoms and minimize side effects. So it's very good, and we still do it a lot. And I suspect we will continue to do it a lot, but it has drawbacks. So many of us, and myself included, for decades now, have recognized that the ultimate answer for Parkinson's will be some sort of biologic treatment. And the most obvious candidate for that, at least back in the 80s, we thought was something called cell transplantation. This is taking brain cells from another source. And you can get cells from patients' own bodies that are sort of similar to dopamine producing cells. You can get them from something called the adrenal cells, which is a little gland that's just above the kidney. You can also harvest dopamine cells from fetuses. And you can take these cells and you can transplant them into the brain. And early studies of fetal cell transplants, this was in the 80s and early 90s, that were done in Europe, were very promising. They showed improvement in patient symptoms. Everyone got very excited. There was a bit of an ethical conundrum because to get fetal cell transplants, you have to get them from aborted fetuses. So there's a bit of an ethical issue there in some countries. But despite all these barriers, there were 2 trials that were actually done in the United States, and these were placebo-controlled studies. So if we have, excuse me, a new biologic treatment, such as a stem cell replacement, and we want to really show that it works, what you have to do is what's called a placebo-controlled trial. And we're going to talk about this more in a few minutes, but you basically have to take 2 groups of people, and one group of people actually gets the cell transplant. The other group of people does not. They get what we call a sham procedure. They get put to sleep. They have an incision or 2 on the head, but we don't actually put cells into the brain. And the patients don't know which group they're in. The caregivers don't know. The neurologists that are doing the evaluations don't know. And then we see if there's really a difference between those 2 groups. And lo and behold, when these 2 placebo-controlled trials of fetal cell transplants were done in the United States and done under very controlled circumstances, there was no difference between the placebo group and the patients that actually got the real transplants. And more importantly, of the patients that did get the transplants, over half of them developed really bad dyskinesia, even when they weren't taking their medications. And this was problematic. So this was sort of a big pause on the field of cell transplantation. Now, there's about to become a renaissance for cell transplant. We're going to talk about that at the end. But in the near term, we kind of want to talk about what do we think went wrong? And when you put cells into the brain, the immune system realizes that they're not supposed to be there. So a lot of these cells don't survive. They're sort of killed off by the immune system. And when you put cells that are foreign into the brain, we think that they don't really know what to do. They don't really know how to integrate into the brain. It's sort of like taking a stranger and putting them into a big social group. They don't know anybody. They don't know what to do. They don't interact very well. And these may have been some of the reasons why these trials did not show more benefit. So around the time that we were realizing we had more work to do with cell transplantation, we started to explore a different biologic approach called gene therapy. And this has really been the focus of my work and the field of sort of biologic surgery for Parkinson's, that field for almost 2 decades now. And gene therapy uses a technique where we take a virus. Now, viruses kind of, everyone's now very aware of viruses. They have a bit of a bad reputation right now. Not all viruses are bad. And there are actually benign viruses that you can use to carry a gene into the brain. And, genes are part of our DNA. And genes are basically instructions. They're basically recipes for telling the cells of our body how to do stuff. And in certain brain cells, the genes are turned on that tell those cells how to make dopamine. So I think about genes as recipes. They're recipes for telling our skin cells to make hair. They tell our fingernail cells to make fingernails. And they tell some of our brain cells to make dopamine. So viruses have DNA as well. This big sort of funny looking structure is what a virus looks like. And when it gets into the body, it latches itself onto a cell in the body. This big blue thing is a cell. It latches onto a cell, it puts its DNA into the cell, and then that cell's function is altered in some way. And usually with viruses, it's usually a bad thing. So for example, the common cold is caused by a virus. It latches on to the cells of your respiratory system. It causes a cold. You have a cold for a couple of days or a week. The cells turn over, the viral DNA disappears, and you get better. So they're very effective machines for getting genes into our body. And what we can do is we can take a virus, get rid of the bad DNA, put an engineered piece of DNA, a good piece of DNA a good gene that we want to teach the cell a new trick. We can then put that modified engineered virus into the brain, get it to latch on to brain cells, and put that good gene into that cell and alter its function in a meaningful way. So there, fortunately, is a naturally occurring virus that occurs in nature. Many of us, all of us, are exposed to it. It's called adeno-associated virus. That was a picture of it. The short lingo name for it is AAV2. And this is a virus that occurs in nature. It does not cause disease in humans. We don't really know how it originated. It's probably a very old virus that maybe caused disease in the past, millions of years ago, and now doesn't. But it has some interesting properties. It doesn't trigger an immune response in the human body if you put it into the brain or the central nervous system. That's what CNS stands for. And it loves brain cells or neurons. It loves to latch on to neurons. So it has a strong affinity for latching onto them if we deliver it to the brain. Now, we started doing trials in Parkinson's patients using this virus. And we looked back to the cell transplantation trials and realized that for different types of transplantation trials, this is fetal allograft. That just means a fetal transplant trial. When you do it in an open label trial, that means everyone's getting the stuff and everyone knows that they're getting the stuff. Everyone seems to get better. But when you do a placebo-controlled trial, like we described before, where half the patients really get a transplant, half don't, and they don't know, you can't demonstrate benefit. And this was true for fetal transplants, for pig dopamine transplants, and even infusion of what's a substance called a growth factor into the brain that we were hoping would promote the survival of neurons. All these looked great in open label trials, but none of them showed benefit in a placebo-controlled trial. Now, in gene therapy, why might this be the case? Well, there is this very strong placebo effect in Parkinson's disease in particular. So if you're a Parkinson's patient and you're taking a medication or you're doing a therapy and you think it's going to make you better, it really can make you better for brief periods of time or sometimes even for extended periods of time. And so we know that this is an issue when we try to design trials for Parkinson's disease. As a surgeon, I think about, how are we delivering this stuff? Are we doing a good job of delivering the gene? And it turns out that in these early trials, we really weren't doing a very good job. This upper right picture is a picture of a gene therapy infusion happening in a real live patient. So you can just barely see the top of the head. You see this kind of bump is the top of the head. There's two little infusion cannulas that are going down into the brain. They're being directed by this very fancy device. We call it a stereotactic frame. This is the device that we use to deliver deep brain stimulating electrodes to the brain, for example. And there were a few patients in one of these early trials that passed away. One of them had an accident, the other had a heart attack after their procedures were done. So these were unrelated to their procedures. And we looked at how much of the gene really got into the part of the brain that we wanted to get the gene into. And it turns out that it was a very small percentage of the target structure really got affected by this virus. It was about 14% of the target. We wanted to get about 50%. We wanted half of this structure to be affected by the virus. We wanted to get the new gene into half of this structure, and we only got about 15%. So we started to think about how we could make the delivery methods better. In the early 2000s, there was one gene therapy trial that we did, and this was a virus, this AAV virus that we're talking about, carrying a gene for something called amino acid decarboxylase. So this gets a little technical, but basically this is a little enzyme. It's a little chemical that your brain uses to convert the precursors of dopamine into dopamine. So we use AADC naturally to make our own dopamine. And when you, as a Parkinson's patient, take carbidopa-levodopa, this is the little enzyme or the little chemical that converts that to usable dopamine in your brain. And as Parkinson's gets worse, the amount of this enzyme drops. And so we think that that's one of the reasons why medications become less effective as Parkinson's advances. You have less of this AADC. So when you take your Sinemet or your carbidopa-levodopa, your brain doesn't do as good a job of converting it to dopamine that your brain can use. And it turns out that there are primates, monkeys, species of monkeys that live a very long time, and as they get older, they can develop Parkinson's-like symptoms. They can get tremor, they can get slowness of movement. You can actually give them a chemical that sort of accelerates that process. And then if you treat them with carbidopa, levodopa, or the medications that you take, it will make them better, just like it does us. We took some of these older monkeys that had developed Parkinson's symptoms, and we infused this AAV gene carrying this gene for amino acid decarboxylase. And lo and behold, those animals got better. Their symptoms got better. We could get special MRI scans of these animals and see that their brains were expressing this gene. And they didn't appear to have any side effects. So it appeared to be a safe treatment. And if you look, these are actually monkey scans. So this is a sort of pre-operative monkey scan. And you can see on the normal side, there's a bright red dot and there's a lot of orange stuff. This is in part of the motor circuit of the brain, but there's very little activity on this other side. And when you treat them with gene therapy, you can see both sides look equal, which means that there's a restoration of expression of this enzyme. And these animals improved clinically. And some of these animals have lived out to 8 years now, and they get special MRI scans where you can see if they're making dopamine. And the expression of this new gene appears to be stable out to many, many years after you initially administer it. This was work done by one of my former colleagues at UCSF, Chris Bankovich. He's now at Ohio State, and we still collaborate a lot together. So we did a phase 1 study using this technique. Now, phase 1 means that everyone's getting the stuff and everyone knows they're getting the stuff. And we use this sort of traditional technique where we use this special frame to put the cannulas in. This is what one of the cannulas look like. It's just it's basically a long, thin needle with a little glass tip at the end. This orange thing is the structure in the brain where we want to infuse the virus. We put 2 cannulas in. The name of that structure is the putamen. We'll put 2 cannulas in, and we push the fluid in using a constant pressure. It's an infusion pump. If you've ever been to a hospital and you have an IV and they use a little pump to put fluid in, that's what the same technique that we use. And it pushes this virus into the structure. The virus latches on to the neurons or brain cells, puts the little gene into them, and then those cells start to do a new function. In this case, those cells start to express or make amino acid decarboxylase. And that's, again, what the picture looks like intraoperatively. So these patients got better. This is a score of severity of Parkinson's symptoms. Higher on the scale is worse, lower on the scale is better. So if you look at their scores before the procedure and after the procedure, everyone had a downward trend. A downward trend in this case is good. They had about a 30%, a little more improvement in their movement symptoms. So this was very encouraging. However, this was a phase 1 study. So we know that phase 1 studies, patients tend to look good. So we knew that at some point we had to take this to a phase 2 study, meaning one group gets the infusion, another group does not. These were the non-invasive, these are these special MRI scans. They're really called PET scans of these patients. In Parkinson's, we sort of retain dopamine production in the front part of the brain here, but there's this little back part that starts to fade away. And you can see after 6 months, after the gene therapy infusion, this back part starts to really light up. It almost looks completely normal in these patients, which is also very encouraging. So we wanted to try to make our delivery techniques better. We were thinking back to that one study where we only got 15% coverage and we wanted to get 50% coverage. We wanted to try to make things less invasive, more efficient. And we started to think about how helpful it might be to actually be able to visualize these infusions in real time. This is the surgical plan for one of the early studies. And this is very complicated. This is 8 different needle passes into the brain, 4 on each side, to try to get 50% coverage of this structure called the putamen, which is part of the circuit that's involved in Parkinson's disease. This is very complicated. This is invasive. This is a lot of penetrations of the brain. It takes a very long time, and you really can't see what's happening. And it was this trial that resulted in 15% coverage of that structure, despite this degree of complexity to try to cover as much of it as possible with multiple needle penetrations. So my partner, Chris Bankovich, made an interesting discovery. So this is a time lapse of one of his monkey surgeries. So this is an elderly monkey that's got Parkinson's disease. We're doing gene therapy, and he realized that if you mix what's called an MRI contrast agent. So contrast is a fluid that shows up on MR scans. If you mix contrast with the gene therapy and then deliver it, you can actually see the infusion happening in real time. You can actually see where the infusion is going. And we thought that this would be very useful to prevent us from doing a procedure on somebody and only getting 15% coverage with future trials. And we found in these monkeys that the area where you see the contrast on the MR correlates very well with the part of the brain where the virus goes. So it's really a what you see is what you get technique. Now, around the same time, another colleague at UCSF and I were developing a technique for placing DBS electrodes, deep brain stimulation electrodes, using MRI guidance. So we started to do these procedures inside of an MRI scanner, and would get pictures during the procedure to guide the electrode placement so it would go right where we wanted it with a very, very high degree of accuracy. And this is a system that is now a commercially available system. It's called ClearPoint. It's a plastic MR compatible device that we temporarily mount on the top of the head after the patient goes to sleep. We can aim that device at a very specific target in the brain wherever we want to put a device, such as an electrode or an infusion cannula. We can get MRI pictures as we're placing the device, so we can make sure it's going right where we want it to. We can monitor to make sure there's no bleeding. And Chris decided that he was going to start using this platform to deliver gene therapy. He designed a ceramic cannula, so it's safe to be in the MRI scanner, and it's very effective at delivering gene therapy to the brain. So this is what it looks like. This is a regular standard MRI scanner. There's a patient in the scanner. You can't see them. There's a little infusion pump here that the virus is loaded onto a syringe on that pump. And then there's a sterile infusion line that's running sort of from the pump into the patient's head in the middle of the scanner. That's what it looks like. That's the top of the patient's head. This is that plastic device that's temporarily mounted on the scalp. This is the ceramic infusion cannula that's going in. And this is what it looks like. So this is a human getting bilateral, that means both sides, infusion of gene therapy, and we're infusing it mixed with this contrast agent, so we can see exactly where the infusion is going in real time. So this was the first time that we've ever delivered a biologic and done it in a way where we can actually monitor the infusion as it's happening. And it turns out that when you can see what's happening, it's really helpful. So one thing that we discovered very quickly is that there's this phenomenon called perivascular spread. And there are lots of little blood vessels in the brain. And the blood vessels are surrounded by a little tiny space of fluid. So it's a little sort of tunnel that's running through the brain. And in the tunnel, there's a blood vessel, but there's a little fluid space between the vessel and the brain. And it turns out that these spaces are a great conduit for vector to be carried away from the intended target. We call this non-targeted delivery. That means the vector is not staying where we want it to be. And I'm going to show you an example of that. So on your left, there's an infusion cannula in the brain. You're going to see an infusion start. It's going to look bright. And then fairly quickly, you're going to see this little thin streak of virus that's being carried away from the intended target up to this other area called the caudate. Now, the spread of this virus around this blood vessel, it's not going through the blood vessel, it's going around it. This spread is not dangerous, but it's also not helpful. The more that this happens, the less the virus is going to stay where we want it to stay, and it's going to be less effective if it's not staying where we want it to stay. I'm going to show you a more dramatic example on your right. We're going to start an infusion, and you're going to see that it very quickly is going to start to spread all over the place. Multiple tiny little blood vessels that we can't even see before we do this on the MRI scanner. You can see it's just spreading all sorts of places. Now, this has been happening all along. This was happening in our early studies when we weren't using this technique. We were simply putting the cannula in the middle of the target. We were assuming that the fluid was just going to fill up the middle of the target, sort of be soak up like a sponge evenly in all directions, a nice little round ball, and that it would behave itself. And it turns out that it doesn't work that way. And this is why we were only getting 15% coverage in these early studies. There's another phenomenon that we suspected was happening, and indeed it is. It's called reflux. And this is where virus starts to leak up around the cannula itself. So I'm going to show you another video on your left. This orange line, I've drawn it at the top of this round structure here. This is the putamen. That's where we want the virus to stay. So we're going to start an infusion, and it's going to look good initially, but then vector is going to start to leak up around the cannula. It's just basically traveling up the path that the cannula has taken through the brain. And all of this white stuff above the orange line is just not helpful for the patient. It's not dangerous, but it's not helpful. And this is why we were getting less coverage on these original procedures than we believed we were. So we've changed our thinking dramatically, the way we design these clinical trials. We've learned that sort of just putting the cannula in the middle of the target, the brain structure is bad, because if you start to get leakage around these blood vessels, the only thing you can do is try to move the tip of the cannula away from those spaces. And if you're already in the middle of the structure, you don't have much place to move. So we've started to infuse sort of at one end of the structure and advance the cannula as we infuse, so we can sort of shape the infusion, and we can also limit the spread from perivascular spaces. We do what we call a stacked infusion. So we put a cannula in, we'll make an infusion, and then we'll advance the cannula and make a second infusion deeper. So that's what we call a stacked infusion. This is what it looks like in a real person. So on your left, we're going to start an infusion. It's going to be pretty well behaved. We're going to start to see a little bit of reflux, and then we advance it and do a deeper infusion. So that's what we call a stacked infusion. The other technique is what we call a progressive infusion. And this is what we tend to use now the most. We sort of start at one end, and then as the infusion is happening, we manually advance the cannula. And I'm going to show you that now on your right side. Again, this is the structure. It's called the putamen. We're going to start infusion. We're going to start to see a little leakage, a little bit of reflux start right there. As it does, we're going to start to advance the cannula. You'll notice that there's very little perivascular spread here because we keep the cannula moving, so we don't give it a lot of time to leak out of those blood vessel spaces. So we've gotten much more efficient. And this technique has also allowed us to deliver more vector. So early on, we learned that we were probably being far too conservative in the amount of virus that we were giving. We did this sort of based on these studies in monkeys, and we were simply sort of scaling up the size of a monkey brain to a human brain, and then sort of scaling up the volume. But these structures, this relative size of these structures is different in a primate or a monkey versus a human, and you can't think about it that simplistically. So by seeing what we can do and seeing how much of the structure we're actually covering, we've been allowed to give much higher volumes. And we think that will increase the effectiveness of these therapies. So there are clinical trials underway using gene therapy. These are happening now all around the country. We're in a very exciting time. A lot of these companies that you see listed here are involved in gene therapy for Parkinson's. They're also involved in gene therapy for other neurodegenerative disorders like Huntington's disease, for example. But the other really exciting thing about some of these companies is that they are about to unleash a number of cell transplantation trials, again, for Parkinson's. So it's been 20 years. The work on cell transplantation has not stopped. It simply has gone back to the drawing board, trying to understand, and we think understand the limitations of the trials that were done in the 80s and 90s. And there are some very exciting new cell transplantation techniques on the horizon. And I think some of the most exciting ones involve harvesting your own cells. And it can be something as simple as a skin biopsy, taking those cells, modifying them in the lab to teach them how to make dopamine, and then giving those cells back to that individual. And the exciting thing about this is that these are your own cells. So they should not trigger an immune response like the original early stem cell transplantation trials did. And again, there are a number of companies here that are actually listed that are actually actively doing trials. So Blue Rock is one of them. And there are other companies that are about to come on the horizon. These are companies like Aspen, like Fujifilm, Cellular Dynamics, a number of them. So it's a very, very exciting time. I really think that we're on the cusp of a very exciting time for biologic treatments in Parkinson's. And maybe next time we get together and talk, we'll have some data to show from these early stem cell transplant trials. It'll be very exciting, I think. So that's all I have. I tried to leave about 20 minutes for questions and answers. I'm going to stop my screen share if I can. Let's see, did I stop it?
You did.
Okay.
And that was extremely fascinating and very cognitively stimulating. So I'm like, wow, this is deep. So I do have a few questions, and I know there's still some coming in. So I'm going to try to go back to some of the ones that's come through, and then hopefully we can catch up with the ones that are now coming in. So one of the questions that I thought was a great one, since you were talking about the AADC, is that something that can be measured to determine progression of Parkinson's?
That's a great question. That has not historically been something that we've actually measured for progression. We do know that the levels of AADC drop. And, I showed a couple of PET scans. I showed, I think, one PET scan in a human where you can see there's less AADC activity before gene therapy. There's more after. Doing before and after pictures is useful because you can see a relative change in that particular individual. But just to sort of get a random picture of a patient, those scans aren't, we don't really know how to quantify these changes to sort of try to measure progression. So it's an interesting idea. Theoretically, you could do that, but it's hard to quantify it. So I don't know that that would be helpful. I might bring that up with our imaging people, though. That's an interesting idea.
Absolutely. Okay, so I've had several questions about this. You talked about this being current studies, and I'm assuming are they in all phase 1 or some of them in phase 2? Like, where
Yep.
are we with that? Because the next question is, if they are enrolling, are there age limitations, or how long someone's had the diagnosis of Parkinson's?
Yep. Yeah. So we're in this kind of weird phase, right, or period of time, I should say. So the sort of two predominant programs, if you will. One of them is this AADC that actually was in phase 2. That study was paused because there were some unexpected MR findings that appeared to be asymptomatic, meaning that the patients that actually were in the treatment group that received the gene therapy seemed to be doing well, didn't seem to be having any adverse effects, but some of the MR scans looked a little funny. So that study got put on hold until we kind of get follow-up MRI scans in those patients. And I don't know what's going to happen with that study. We actually, I think, have a call in a couple of weeks to get an update from the sponsor. So that was in phase 2. The other predominant program for Parkinson's is a gene therapy called GDNF. GDNF is a growth factor. You may have heard other speakers talk about it. It's a growth factor that helps promote survival and health of neurons. The exciting thing about that trial is that the hope is that it can stop the progression of Parkinson's disease. And that's just completing a phase 1. And what happens is you do a phase 1 study, everyone gets the stuff. Generally speaking, everyone gets better. You look at the safety. And when I say you, I mean the FDA looks at the safety. They make sure that there's nothing unexpected, no side effects. And then if it looks good, it goes to a phase 2. So GDNF is just finishing phase 1. There'll be a pause of probably, I'm guessing 9 months, and then it'll probably start a phase 2. So that's kind of where we are with gene therapy for Parkinson's. There's the Blue Rock group is doing, the company that's the sponsor, Blue Rock, is currently doing a phase 1 cell transplantation trial. And then there's one also going on in Europe called TransEuro. So that's only for European centers. So yes, these trials are happening. The best way to sort of, in the US, I think, to keep an eye on these things is to go to, if you've never been, clinicaltrials.gov. I don't know if you guys are familiar with that. But if
I actually just asked Kelly to put that up.
Yep. So clinicaltrials.gov, all one word. If you are a US sponsor or you're a US investigator and you're doing a clinical trial, you are required to register the trial on that website. And you can search terms, you can search acne, hair loss, Parkinson's disease, whatever, and it'll pull up every trial that's being done. So it's a great resource.
Fantastic. All right. We have lots of just really, really good questions. And I want to go back to a comment we received from, I have to scroll up here, Edwin, I think it was, who said that he was part of your trials. And
hi, Edwin.
Yes, 6 years ago, number 16, and can report great benefits in regards to movement and decrease in dosage of meds. Which then leads me to another question. Someone had asked about the impact on non-motor symptoms. So we see maybe some obvious motor symptoms, but what have you seen in terms of non-motor?
Yeah, so that's an excellent question. So, you know, we always talk about the motor symptoms of Parkinson's: tremor, slowness of movement, stiffness, shuffling gait. There are many, many what we call non-motor symptoms: constipation, memory troubles, lightheaded when you stand up. I mean, the list goes on and on. Trouble peeing, trouble sleeping. One of my mentors used to say, if you want to make patients happy, help them go to the bathroom and sleep. If you can eat, sleep, and go to the bathroom, that's the three secrets of life. And Parkinson's patients have a lot of trouble sleeping, and many people have trouble going to the bathroom. So these are important symptoms. We certainly are looking at the outcomes in these non-motor symptoms of Parkinson's. Cognition, for example, is something that we track fairly closely. We don't see any worsening, but we don't see a lot of improvement. And this may be because, Parkinson's involves dopamine loss in the movement part of the brain, but there is also dopamine loss in other parts of the brain as well. And when we do these very directed therapies, we're doing them sort of at the motor circuit or in the motor circuit. And so I'm very interested in seeing if the growth factor trial actually helps non-motor symptoms. And the reason for that is in that trial, we're actually putting the GDNF not only in the motor part, but we're also putting some of it in the non-motor part of that structure, the structure, the part of the structure that may be more involved in memory or cognition, for example. So the jury is still out on that.
And we did have a question earlier too, in terms of doing these kind of studies. If somebody's had DBS, does this make them ineligible for this kind of a trial, or potentially this type of treatment if it, gets to market? Yep,
excellent question too. So the FDA is pretty specific about how they allow us to construct these studies. And we're trying to convince them and the scientific community that the intervention that we're doing is really making the change. And DBS is something that really improves symptoms. And sometimes just the act of putting the electrodes in can make the symptoms better, even if the device is not on. So if you want to participate in one of these early phase 1 or phase 2 trials, at least currently, you can't be part of them if you've had any other brain operation, and that includes DBS. Now, the beautiful thing, the reason we like DBS is that it's non-destructive. It doesn't really physically alter the circuit. So as these therapies become approved, then anyone can get them. So if you want to be in a study, DBS would preclude you from being part of the study. But once these things become approved therapies, then they would be open for anybody. And I personally think that I don't think that DBS is going to go away. DBS is very effective at treating the symptoms we already have. But GDNF, for example, let's say that is does what we hope and pray it does, which is that it slows the progression of the disease. You could have DBS to treat the symptoms you currently have, also get a little GDNF gene therapy that would slow the progression of your Parkinson's. We actually wrote a paper making an argument that it makes sense to actually do these trials in patients that have DBS because you could turn it off and it's like it's not there. But we've convinced the FDA of a lot of stuff. We haven't convinced them of that yet.
Well, I guess one of the questions that just came in would be a great follow-up to that is someone wanted to know, would you expect that there will eventually be a single approach to treating PD, or do you think the precision medicine will end up being many different approaches for many different types of PD?
Yep. Yeah, that's a very astute question. These are good questions. You got a good group.
Yeah, very smart people.
Here's the frustrating thing about Parkinson's. There's this, we all know that part of the brain, it's the substantia nigra is dying off, and those brain cells are projecting their little output to the putamen. That's the target for most of these therapies. So that pathway, we call it the nigrostriatal pathway. That pathway is what's degenerating in Parkinson's. And our brains are pretty remarkable. They've got a lot of redundancy built into them. So, we're constantly losing brain cells. We've been doing it all of our lives. And you really don't notice it until you really get into your, sixth and seventh and eighth decade. I mean, my dad's 102 and he's still pretty damn sharp. But if you get an MRI scan of him, which I've actually seen a scan of his brain looks like a little raisin. I mean, he's lost a lot of function. We don't all, turn into nonsensical people when we get into our 70s and 80s because we have or beyond, because we have a lot of redundancy in our brain. So by the time that you develop symptoms of Parkinson's disease, about 80% of that pathway is already gone. So, if you're going to give a growth factor treatment, what you'd really want to do is detect who's going to get Parkinson's before they get it and actually treat them before that degeneration starts. And if you look at research that the NIH funds, and you look at the research that other groups like Michael J. Fox, for example, they really, there's a huge effort on early detection. How do we early detect people even before they become symptomatic? So I think that these therapies are going to be successful, but the technology that we currently have, we're going to be employing them in people that are already symptomatic and a bunch of their brain is already degenerated. So I think in the near term, it's going to be a combination. We're probably going to be using a combination of DBS and gene therapy, maybe a combination of DBS and cell transplantation, maybe all three. At some point, eventually, when we can predict who's going to get it before they actually become symptomatic, then I think things may change. But I think we're about to enter a phase where it's going to be a multi-modality, a multi-therapeutic approach. That's my guess.
And we had a question about the studies. Are they interested in families with multiple family members with Parkinson's, 2 siblings, a father, maybe history of a deceased paternal grandmother, as well as, other like cousins and things like that? For those kind of studies, are they also interested in people who have a real strong family tie to Parkinson's?
Yeah, that's an interesting question. I mean, most cases of Parkinson's are what we call sporadic or idiopathic, meaning that there's not a genetic component to them necessarily. There's not a specific, mutation. It's just sort of random bad luck. But there are forms of Parkinson's that are familial. There are forms of Parkinson's that are associated with a very specific genetic mutation. And it's interesting that this question has come up. In the past, the studies really haven't focused on that, but we just got a grant. And when I say we, so Chris Bankovich has been my sort of basic science partner for many, many years. Another basic science partner of mine is a really brilliant scientist as well. His name is Jeff Cordover. And Jeff has now just moved to Arizona State, which is right up the road from where I am. And we just got a grant to look at a novel therapeutic approach in a very specific type of Parkinson's that has more of a genetic component. So we're starting to focus more on that. The goal is to try to help everybody. But these very specific types of Parkinson's and these very specific contexts may be very, very instructional to sort of understand the success of these therapeutics. So there probably is going to be more interest moving forward.
That's fascinating. Someone was really interested in, and you mentioned it earlier, the placebo effect. So they're wanting to know, and I'm going to find my question now. Are there any trials on the placebo effect, how we can trick ourselves into improvement? And really, how long does that placebo effect last when you see it?
Yeah, so the placebo effect, it's not necessarily a 24/7 effect. There's always the example of, the father with Parkinson's has daughter that's getting married. And even though they really have trouble walking, they can sort of pull it together and walk the daughter down the aisle. That's sort of the classic story, and then they look like hell at the reception, but they can kind of pull it together for brief periods of time and actually have pretty good function. And we think that's what's happened. That's one of the things that happens in these studies. Patients they go through periods in a study where they'll fill out diaries or they'll come in for an evaluation and they look really good and they report that they're doing really well. They may not be that way 24/7, but they can be intermittently really good for a year, year and a half. I mean, it can be a very prolonged amount of time. So it's not necessarily a bad thing, but it's a challenging thing when you're trying to get a new biologic approved, by the FDA. I mean, if you say this works, but the group that gets it is really equally improved to the group that doesn't get it, that's not good scientific evidence for the FDA to say, well, okay, we believe this really works. So it's a big challenge when we are trying to design these studies.
We had interest in knowing what gene mutation is your study going to be on?
I knew that was going to come up. There's going to be a press release, and so it hasn't been announced yet. Okay. And I don't want to. Jeff would drive down here and cut my head off if the grant has been awarded. It's going to happen, but I think we're not quite ready to announce it yet.
And earlier, someone said they were in a stem cell phase 2 study in Houston. And if you had any further comments on that study?
I don't know which one that would have been. It's a little, yeah. I mean, there have been. I don't know if it was a new one or an old one, but the one that's happening currently is a study called Blue. It's sponsored by a company called Blue Rock. It's very exciting. Again, this new wave of stem cell transplants, it's really sort of taking your own cells and sort of teaching them to be, what we want them to be, and then putting them back, basically. It's very exciting. What works in the lab doesn't necessarily work in nature. So we do have to be cautious. We've had a lot of excitement and then had disappointments. But I do believe that, we are going to find a cure for Parkinson's. Parkinson's is actually a perfect disease for a cure. We know what cells are being lost. We know where they are. We know what they do. It's very, very different than diseases like Alzheimer's disease, for example, where it's the entire brain is sort of going bad. In Parkinson's, it's actually a fairly discrete area. And it'll happen in our lifetime. It's just a matter of who's going to do it. We're going to try.
And we're all very thankful that we have folks out there like you, the experts that are trying and doing these very fascinating studies. So I appreciate that. I know everyone here does, and you explaining it so well. I know we're just about done. I am kind of curious as too. So when someone's doing these studies or these treatments, are they a one-time procedure, or is this something they would do serially?
Really good question. Yeah, I was just looking through the chat. You've got an amazing group. I mean, we got CRISPR questions, and
I know I didn't even get to that one yet.
LARC2, GBA. I mean, it's just like really, it's a phenomenal group. So right now, these are one-time procedures. And for example, in gene therapy using AAV, we've got, again, we've got primates that we've treated up to 8 years and there's stable expression. And there were patients in our first AADC study in San Francisco that had PET scans for years afterwards, and it appears to be stable expression. So right now, these are all, it's a one and done procedure. And that's part of the exciting thing of it as well. DBS, as good as it is, there's ongoing stuff. Batteries have to get replaced. If a wire breaks, you have to replace it. So this is a one and done, which is very appealing.
Absolutely. Dr. Larson, thank you so much this has been extremely fascinating, and I really appreciate all of your information, and we have had some amazing questions. So thank you everyone for your fantastic questions. So we have a little thing that we always do as we thank our speakers and of course show gratitude towards you as well as to be together. And that's our wave of gratitude. So if everyone would be willing to share the wave of gratitude to Dr. Larson and thank him so much for this fantastic presentation and conversation. And again, thank you everyone for joining us. We're so glad. This is a great way to kick off 2022 with such great hope. So thank you for that.
Great. Thank you, guys. Amazing group. I really appreciate the opportunity. Come see us in Arizona.
Absolutely. And for those of you who want to know, I didn't see the question come through, but this is being recorded and we will have slides available to you in a couple of days. So thank you so much.
Thank you. Have a good day, everybody.
Bye-bye.